Hello and welcome to Brian's Backyard Biology. Today what we're going to be doing is talking about interpreting cladograms, which cladograms like we're going to see here, it's kind of looking at the evolutionary family tree. So in this particular unit we've been talking about evolution a lot and some of the different aspects, the scientific aspects of what evolution is and what it tells us, why it's important. And today we're going to look at these family trees, and these really are just diagrams that help us to see the evolutionary relationship between living organisms. So phylogeny is the evolutionary history of an organism, and that's going to be represented on these diagrams called cladograms. And so you can see here in this particular example, we have this branching diagram that we see. And this branching diagram here is showing different types of species or different types of organisms. Remember, a species is a group of organisms, right, that can interbreed and produce fertile offspring. So when we say that this is species A, that doesn't necessarily mean that it is one organism, but it is a species. It could be a group of species. There could be 1,000 different organisms here. This, A could be zebra, for example, okay? B could be horses. So we're talking about vast numbers of organisms that could be there. Now, we can get down to a specific species. Sometimes we can even get to a subspecies. What we won't have on these cladograms at the end are going to be individual species. And that's really important for us to understand, because remember, like we said in earlier presentations, said that populations evolve, individuals don't evolve, right? And that is one of the most difficult concepts for us to understand, is that it's the actual population that's changing, not the individual. And I'll give you some examples of that here in a little bit. All right, so understanding and being able to read these cladograms. So the first cladogram that we saw kind of had this, kind of a branching pattern to it. Here we have something that looks a little bit more, I think looks more like a field goal, the goalpost in a field goal. But really they're saying the same thing. Here we have A and B that branched from each other. If we look at that first one, we have A and B that branched from each other. And so these really are the same cladogram. And really we need to just understand kind of how they're read, right? So we have a couple of different things here. First of all, notice where the ancestors are in the present day. This is showing that we're reading in this direction. We're reading from the bottom of this cladogram up to the top or the ends where those species are indicated. Those species that we see on the end, these are called tip taxa. Okay, a taxa is a group of organisms, and so these are at the tip of the cladogram, and so that's why they're called tip taxa. When we're looking at a cladogram, in order to understand what it's actually saying is, remember we talked about the speciation events, right? We had things like geographic isolation or reproductive isolation with the temporal or the behavioral isolation. We had adaptive radiation, all of these different types of divergent evolution. Those speciation events are represented by the branching or the split in these cladograms. And so here we can see that the blue line is going to talk about the ancestral lineage, and that just means that it's a common ancestor between those. And then we have that speciation event where it splits. That point where it splits, where that box is showing, that is called a node on a cladogram, N-O-D-E is what that's called. And so when we look at this, what it's telling us is that individual B and individual C have their own unique lineage that's there, but then they also share a common lineage that we see here. And so that's represented by the dotted line. The solid black line would be the unique lineage to organism A that's there. All right, and those organisms or those species are going to have traits that are very unique to themselves. But each lineage is also going to have unique ancestors. All right, so just kind of, if we think about these like family trees, since that's what I said they were at the beginning, we can think about it kind of like our own family tree, right? So two of my friends, one that is a Kerr and one that is a Brockman, they met later in life as adults through marriage. And what they found out was that there is actually a place in Cincinnati, a cemetery in Cincinnati that is called the Kerr-Brockman Cemetery. And this particular cemetery, they started doing some research there, and they actually each belong to, even though they grew up completely separate from each other, they actually belong to the families, if they trace their ancestry back far enough, they belong to the families that that particular cemetery is named after. And so this Kerr family has its own unique kind of ancestors that are there. The Brockman has its own unique ancestors that are there. But at one point, if we go back far enough, we can find that common ancestor between those two families. And that's kind of what we're talking about here. This species C has common ancestors, and those common ancestors are unique to species C. They don't share them with anything else. And B would have its own unique common ancestors. But once we get back to that speciation event, that node, that is the last time that these two species, species B and C, shared a common ancestor. And if we go back a little bit further, the speciation event down at the bottom, that's the last time that A, B, and C all shared a common ancestor together. So where that speciation event takes place, that's also the last common ancestor that those particular species had in common at that particular time. All right, so being able to read a cladogram and identify these different organisms and how they're related are important. And so we talk about clades a lot, and that's where a cladogram came from. It's a diagram of clades. And so a clade is a group of lineages. So that means that they share a common ancestor. So imagine going up to a tree and just clipping off a branch. All of the small little branches that are attached to that main branch that fall off, that represents a clade. All right, so if we can make one little cut, all of the ancestors that fall off of that diagram would be part of that clade. So here we can see on this particular one that we have the pink circle. Anything in that pink circle would be identified as a clade because we can make one cut, and that cut is right down here at the bottom. We cut it once, and everything within that circle is going to fall off at that particular place. We could look at another clade. If I cut all the way down here at the bottom, that whole entire branch would be one clade because it would all fall off. I could actually cut just one of these little sticks, and that would be a clade. That would actually be our smallest clade that we would have, because that little stick, all of the common ancestors, including the ones that are existing right now, those would all be part of that clade that would fall off. All right, so we have a couple of questions that as we go through here that you'll be answering. So your first question to answer is, what is our definition of a clade? And if you need to go back and take a look at it, feel free to do that. So what is our definition of a clade? How would you define it? All right, now that you have that written down or typed in to see what that is, you should have something that's similar to this, a group that includes all the common ancestors and all the descendants of that particular ancestor. So we look at where the ancestor is and then all of the descendants from that. And that would be considered a clade. So we can think of it kind of like a family group. All right? So question number 2 that you're going to answer, look at the image to the right. And all I want you to do for this one is for the green, the blue, the pink, and the orange, I want you to go ahead and write down either yes or no. Okay? And it is perfectly fine for you to have these and to make corrections as you go. That's completely and totally fine. But really try to answer these questions without seeing what the answer is ahead of time. So go ahead and pause the video and just answer the question. Is it a clade or is it not a clade? So we will start with the green one. And we ask ourselves, is this a clade? So the question is, if I cut this in one spot, do all of the ancestors fall off? And the answer to that is yes. Right? If I cut right here, then we know that all of those organisms fall off. It's not only is it all of the ancestors, it's all of the descendants of those particular organisms. What about the blue one? Again, one cut, and all of the descendants are going to fall off. And so again, this would be an example of our smallest clade that we have. What about the pink? Well, the pink, if I make one cut here, in order to get this over here, I'd have to make a second cut. So those would not be in the same clade. And you say, well, wait a second, I could just come down here and cut the very bottom and both of those would fall off. You're right, they would. But so would this over here on the right, and so would this group over on the left. And so the box here is not including those, and so those are not considered to be organisms within the same clade or within the same group. Right? We would have to include all of them for that. And then lastly, we have the orange one. And your answer for the orange one also is no, because again, we would cut down here at the bottom, and what are we doing? We're excluding this group out here. That would also fall off. If I cut down here at the bottom, this branch over here to the right will also fall off. And so that would have to be included in order for this to be an appropriate clade, okay, or a family group. All right, so reading the cladograms and reading the clades, understanding what those are, clades are going to be nested inside of one another. And so we have this small group here. Maybe this is bats, for example, and then the bats are mammals, right? And so they're inside this blue one maybe is all of the mammals that are there. And then we have those that are the mammals are a vertebrate, meaning that they have a backbone. So that's where the pink one comes in. So this is going to be all of the organisms that have the backbone. And you can see how our groups continue to get bigger and bigger and bigger with more and more characteristics. So they're kind of nested together. So if you've ever seen these little nesting dolls where one doll fits into another one and you can kind of take those apart in half and put them inside of each other and they all stack up and eventually all of these would fit inside that bear, right? And so that's kind of the way that these clades work. But just remember, a clade may include thousands of species, like mammals, or it could just be one species, right? So it's very easy for people to misinterpret cladograms. A lot of people think that when we look at a cladogram or we look at these family trees, that somehow they're becoming more advanced, but that's really not the case. And in fact, when we look at this, I'm going to kind of use this cladogram that we see here at the bottom as an example going through, but we see mosses, fern, pine, and rose. And a lot of times people look at this and say, oh yeah, the moss over there, they're just these teeny tiny little things and they're not very complex at all. They must be less advanced than a pine tree or a rose because a rose has flowers, so obviously they are the most advanced. That's not necessarily the way to think about it. When we thought about organisms originally, if we go all the way back to the ancient Greeks and we look at Aristotle and some of these other philosophers, when they came up with kind of their idea of organisms, Darwin wasn't the very first person to suggest that organisms change over time, by the way. He's the one that his theory has kind of stood the test of time, but there were plenty of theories before him that suggested that organisms change over time. They just were flawed for whatever reason. A lot of people thought that there was some sort of ladder of complexity, that things were trying to become more complex as they changed. This has been disproven that it's not that way. An organism isn't striving to become more complex like we thought back in the ancient Greeks, but that idea or that way of thinking still kind of persists even today, and we start to see that. And so when somebody looks at this, they say, well, A must be less complex and D must be more complex. And so in either one of these that we look at, and that's not the case. It's not talking about complexity. And similar, when people look at these, we have learned our whole entire life to read from left to right. From the very first time that we sit down with our parents and start identifying letters, we identify left to right. When you sit down at a computer and you look at a computer screen, the top left corner is the corner that you're going to look at most because we read from that top left corner to the right. So we look at a cladogram and all of a sudden we want to read A, B, C, D. And I even have these letters in sequential order on there, so I'm not doing you any favors, right? And so we want to look at this and we say, okay, well, D must be greater than C, which is greater than B, which is greater than A. And again, that's talking about that kind of complexity, right? That we're becoming more advanced as we move from left to right. And it's just not the case, but that's another misinterpretation of cladograms that is very, very frequent. And the third one that we have is that just that reading left to right, and that tells us how closely related things are. So not that we're going in a sequential order, but we try to associate how closely something is related by its position on the cladogram. And so we're still trying to read that left to right. What we need to understand is that cladograms are read from the bottom to the top. They're read from the ancestor up to the current day species, those tip taxa, right? So we would start down here with the moss, and or down here at the bottom, and we would start to work our way up. And let me get back to where I was. As we work our way up, what this says is that there was a speciation event that took place, and the moss separated from the common ancestor of the fern, pine, rose. Now that common ancestor does not exist on Earth today. It's extinct. It's gone. Okay, whatever that organism was that was living on Earth, and the ancestor of the moss, again, that is extinct and not living, those two separated at this particular time. Well, if we look at this one, it's saying really the same thing, even though the moss is on the right-hand side. We started out with this one common ancestor. There was a speciation event, and the two common ancestors, the common ancestor of the moss and the common ancestor of the fern, pine, rose, together split off from each other. Then we had the common ancestor of the fern split off next. This one's showing the same thing. And then the pine and the rose shared that common ancestor last. And here we have the pine and the rose sharing the common ancestor last. So these two are exactly the same. We're not going to read left to right. So if I ask, and this is a very common question that I ask, and one that you should be able to answer, is what is more closely related? Is a fern more closely related to the moss, or is the fern more closely related to a rose? All right, well, the fern, if we look at this, the fern is actually going to be more closely related to the rose, right? Because if I come down here and I cut this clade, would include all of those. Remember, and they shared a common ancestor there. The moss didn't share a common ancestor with the fern until we get all the way back down here at the beginning. And that goes for this side as well. Here would be our common ancestor of the fern and the rose, and the common ancestor of the fern and the moss would be all the way back at the beginning. And so when we look at these, they're right next to each other. And so a lot of people say, well, it's more closely related to the moss. But if we just rewrite that a little bit, don't change the relationship at all, but rewrite it. Now we have the fern that's all the way on the opposite side. So we can't just look at the tip taxa and read from left to right. We really have to read from the bottom up to be able to understand these. All right? And so when we look at cladograms and we look at this, most people look at that and they say, you know what? That makes a whole lot of sense. And I can see how these, speciation events occur and all that other stuff. And really the problems with evolution that we see that people have, and they start to say that, well, I don't believe in evolution or whatever the case, really comes from humans and how humans evolve. And so remember, we need to remember these kind of fundamental concepts of what the theory of evolution is saying and what it's not saying. Remember that humans did not evolve from chimpanzees. Humans, we can see, are listed here on this particular cladogram. We have the bonobos and we have the chimpanzees that are over here. They actually make up a group that are more similar to each other, and then we have humans off to the side, and then we have gorillas, and these would all be primates, a particular group of mammals. Within the primates, there is another group called the hominids, and the hominids are primates that walk bipedally or walk upright on 2 feet. Bipedal means 2 feet. And so there is only one species that fits into that hominid group, and that hominid group includes the humans. Now, if we go far enough back in time, we can see that there were ancestor hominids, species, that were alive. If we look at the fossil record, they were alive on Earth at one point. They don't exist anymore. They've become extinct, right? So how do all those relationships tie into each other? Well, there's a lot more hominids out there than really probably what you realize. At last check that I had, there were around 17 different hominid species that we found in the fossil record, and only one of those is still left, and that is us, Homo sapiens. So those were our ancestors. Chimpanzees are not our ancestors, right? Because chimpanzees are, if we look over here, our chimpanzees are a tip taxa. That means that they are living today. We call those extant organisms if they're living. Extinct, not living. Extant, they are living. So these chimpanzees are living today. Humans are living today. Remember, living organisms can't change and evolve into other living organisms. I'm not going to get a turtle that evolves into an alligator, right? They're both living at the same time. Now they're both reptiles, so they probably, if we follow that back far enough, we can find a common ancestor, right? But turtles aren't turning into alligators, and it's the exact same thing that we're saying here. We're also saying that humans aren't necessarily more evolved. We're not more advanced than everything else. We're just different, and it's okay to be different when we look at these things. We don't have to be the most advanced thing to somehow be better than everything else. We obviously have a perspective from a human, so a lot of times we personify different things because of our own experience with the world around us. But really, we're all just uniquely adapted to the environment in which we live. And so those adaptations serve us very well as humans. Our species has been quite successful in the short period of time that it's been on Earth. So how is all of this relevant? Like, who cares about all these cladograms and everything? So I decided to bring this in because this was actually a talk that I was listening to from a researcher at Ohio State. And I'm really hoping to be able to interview this person. Her name is Dr. Linda Safe. She is a world-renowned virologist, and I just happen to know her because she's one of my high school friends' mothers, and I think that that's really, really cool that I knew her growing up before she became so world famous and discovered all these things about viruses. But the interesting thing is, for the past 30, 40 years, she's been studying coronaviruses, which is really applicable today. So in her talk that she was giving, this is one of the diagrams that she showed. This is not from her paper, but it's from another paper. And this is a family tree of coronaviruses. So these viruses are a clade. They're a family of viruses that are out there. And you can see that here's our cladogram. We have this big, huge clade. And while you may not recognize all the different names here, that's completely and totally fine. I went ahead and highlighted over here where the SARS-CoV-2, that's the one that's causing the coronavirus, okay, or the COVID-19 disease that we're experiencing right now. This is the virus that's doing that. So what is it most similar to, right? So there might be some things that you're familiar with. The first one, the SARS, if you heard about that back in 2003, there was a virus called SARS that was around, and we see that SARS starts showing up here. It's even the SARS-CoV-2. And so we have several of those different types of viruses. But we also have different bat coronaviruses that are here. And bats are notorious for these coronaviruses as a kind of a breeding ground for those. So why is this important, though? And why do we make these cladograms? So that we understand this relationship between organisms and that they could possibly change, and then we get new species, right, that form, or new viruses that form. This really helps us to drive our research and our vaccines and our treatments and everything like that, because you'll notice up here, this is talking about one polypeptide or one particular protein. And so if I go ahead and exit my presentation here real quick, I'm going to show you, this is the actual paper that this comes from, this study. And this study was just done this year. And so what we see here is, this is all of these numbers and that we have. This really is showing us the relationship between all of the different proteins of that virus and other viruses that they have in common. So you'll if you listen to the news or anything like that, a lot of the researchers are trying to figure out where exactly did this virus come from. And that's an important question for us to really ask, because if we can figure out where the virus came from, then we can try to minimize the exposure there, right? So if it's coming from, we know that it's being transmitted just from a particular organism, and we can figure out what that organism is, well, we stop that interaction with that particular organism. Now, the thing with the SARS-CoV-2 or this COVID-19 virus that we have is we're starting to see, and we've seen now for a while, this community spread, okay, where it doesn't need that animal host that we had before. Now it's going from person to person, and so that makes it a little bit more difficult. But we still can learn if we understand what virus it came from, where that speciation event took place, and what it's most common to. If we have treatments for that other virus, then that means they're more likely to work with this one. And so what we do is we look at this, and so this is looking at a bat coronavirus, and then this is looking at the SARS. Okay, so this is the second SARS virus that we have. And so if we look at that, these are percentages. And so you can see percentages that they have in common with this particular bat coronavirus, and then percentages that they have in common with the SARS coronavirus, right? And so how do they come up with these percentages? Well, they actually plug this into a computer. So this jargon that we have here, all of these letters, is basically we have these two different bat viruses that we think might be similar. We have our COVID-2 or COVID-19 virus here, the third one down, and then we have the SARS that we had back in 2003. And all of these letters represent a specific amino acid, right? So we have things like methionine and isoleucine, the I there. So they're all given letters, and we look at the sequence of those amino acids and how they're put in order. And so this is very similar to looking at a DNA sequence, but we're just looking at the amino acid sequence for those proteins. And we're looking at which ones they have similar. So if they're black, that means that they have those in common with the other viruses. And if they're white, then that means that they're different from the other ones, right? And so we can put these into a computer, and then it will tell us how similar they are, but it's just amino acid after amino acid. You can see that we have up to 541 different amino acids there, and then they looked at other ones. And so this was a very extensive report that we went through in order to understand it. But that just gives you an idea of how scientists are still using this idea, this concept that organisms change over time, and those speciation events that take place help us to understand the life that we have on Earth. And this just happens to pertain to how we treat those infectious diseases, right? And so this concept we have lots and lots of evidence to support this idea that organisms change over time, and it's kind of just the accepted way that we operate at this particular point with regards to evolution. All right, so quick question number 3. We only have a couple questions left. So this one, what is this called? So you should say that it is called a cladogram. And then the second one is, what do you think the red lines, and that says behind my picture there, it says, what do the red lines represent? So what do you think this red line and that red line and that red line, what do all those represent? So those red lines are going to represent the traits that clade has in common. All right, so this is a common trait for that particular clade. So if we were to cut here, that would take care of all of these organisms, and what all of those organisms share in common is that they all are vertebrates. They all have a backbone. The other group that we have here, if we cut here, everything except for the sharks would fall off. So they all have a bony skeleton. If we cut here, then everything except for the sharks and the ray fin fish are in there. They all have 4 limbs, and you can see as we move up, these all have amniotic eggs. These have eggs with shells. These have hair, right? So if I wanted to have feathers, where would you think that I would put feathers? If you said right here after the split from the crocodiles, you'd be right. Okay, we could put feathers there. And then if we wanted to, both of these are mammals, so if we wanted to come up with some characteristic or some difference between primates and rodents, we could probably do that and put that on there. So those would be how you read, again, the cladogram. All right, so creating cladograms, a lot of times we do this using using charts, looking at similarities between these, and that's basically what this is showing, is how we can look at which organisms have particular traits. So which ones have jaws? Okay, so all of these organisms have jaws except for the lamprey, and so you can see the little line for jaws goes right there after the lamprey has already been separated off. The lungs, we have the sea bass, and so they are negative. I didn't have to worry about the lampreys because we already separated those off. So once we separate them off, we can kind of forget about those, right? And so that chart kind of shows us how we do that. And so our last question that we have here, for just to check your understanding a little bit, is the salamander, we have the salamander right here, is the salamander going to be more closely related to the salmon, or is it more closely related to the hamster? Right, so go ahead and write down your answer and see what you think. If you said that the salamander was more closely related to the hamster, you would be right. Now, why is it more closely related? Well, they both have lungs. If we come down here and we look, we can cut right here. This whole entire clade would fall off, and they both have lungs. The salmon doesn't have lungs, it has gills, and so it's separated off before the formation of lungs. And so that kind of gives you an idea of how all these organisms are related. So hopefully that helps you to understand the concept of evolution a little bit better. And some of the different evidence, and not only that, but also how we can use that information, that evolutionary information, to help us solve some problems that we might be facing, like the current pandemic that we're in. All right, so that is cladograms, dealing with evolution. I hope that that was helpful. Just remember to stay safe, wash your hands, stay 6 feet apart, and we'll see you next time.